Welcome everybody. My name is Angelina Lesnikova, and I would like to present you our work entitled Chondroitinase and Antidepressants Promote Plasticity by releasing TRKB from dephosphorylating control of PTPσ in provalbumin neurons. This work was done in the lab of Professor Restrain at the University of Helsinki. Plasticity is the ability of the brain to change itself through establishing new neuronal connections and rewiring existing ones. Plasticity is prominent in early life and peaks during the so-called critical periods, when the ability of the brain to adapt is at its highest. After the end of the critical period, plasticity persists, but at significantly diminished levels. Closure of the critical periods is mediated by changes in cortical excitatory-inhibitory balance that take place due to maturation of cortical inhibitory interneurons. Fast-spiking interneurons expressing parvalbumin orchestrate synchronous neuronal oscillations and play a particularly important role in this process. Closure of the critical period coincides with the functional maturation of parvalbumin-positive cells and establishment of perineuronal nets around. Perineuronal nets are mesh-like structures of extracellular matrix that surround the somata and proximal dendrites of certain types of neurons, in particular parvalbumin interneurons. After the closure of a critical period, neuronal plasticity can still be modulated, and critical period-like plasticity can be induced in the adult brain by a number of different methods. Critical period-like plasticity can be induced by chondroitinase ABC treatment, which digests perineuronal nets, or by antidepressant treatment. Our lab has shown that antidepressants promote plasticity by activating neurotrophic receptor tyrosine kinase 2, TRKB, the receptor for brain-derived neurotrophic factor. More recently, we have demonstrated that activation of TRKB in parvalbumin neurons is both necessary and sufficient for antidepressant-induced plasticity in the mature nervous system. We now propose that modulation of interaction between TRKB and PTPσ, which is a receptor for chondroitin sulfate proteoglycans, a major component of perineural nets, mediates plasticity in the adult nervous system. We have run a series of experiments to test this hypothesis. We tested an idea that TRKB in parvalbumin-positive interneurons is essential for chondroitinase-induced plasticity, and we used an ocular dominance paradigm to address that. We've taken a mouse line heterozygous for full-length TRKB expression in parvalbumin cells, and we used mice with intact TRKB expression as controls. The mice underwent the first session of optical imaging to assess their baseline levels of ocular dominance. Under normal conditions, mice tend to demonstrate the contralateral eye dominance in the binocular region of their visual cortex in each hemisphere. We then closed the dominant eye by suture and injected chondroitinase into the binocular region of the mouse visual cortex. The suture was done to force the activation of the weaker ipsilateral eye. After 1 week of monocular deprivation, the eyes were opened and the second imaging session took place. During the session, the mouse was placed in front of a monitor, and activity of the visual cortex was stimulated by a horizontal drifting bar. The activity was measured using the optical imaging of the intrinsic signal. As expected, vehicle-treated wild-type mice failed to show any ocular dominance plasticity in the second imaging session right after the monocular deprivation, while wild-type mice treated with chondroitinase showed a clear shift in the ocular dominance towards the non-deprived eye. However, mice heterozygous for TRKB expression in parvalbumin cells failed to show any shift in ocular dominance, regardless of if they were treated with chondroitinase or not. These data demonstrate that chondroitinase-mediated reactivation of visual cortical plasticity is dependent on intact TRKB signaling in parvalbumin into neurons. Next, we assessed whether perineuronal net digestion with chondroitinase induces TRKB activation in primary cortical neurons. We treated right cortical neurons with chondroitinase for 30 minutes and checked the activation of TRKB by assessing its phosphorylation levels. We observed that reduction of perineuronal nets by chondroitinase treatment prominently increases phosphorylation of TRKB in vitro. Then, we investigated whether treatment with agrican, a major perineuronal net component in the adult brain, might have an opposing effect on phospho-TRKB. We incubated cortical neurons with agrican in the medium for 24 hours and then treated them with BDNF. Agrican treatment did not alter basal phosphorylation levels of TRKB that are normally very low in vitro, but it significantly decreased BDNF-induced activation of TRKB. Taken together, these data demonstrate that perineuronal nets exert negative effect on TRKB activation in vitro. PTP-sigma is a recognized inhibitor of neuronal plasticity. It is a receptor for chondroitin sulfate proteoglycans, and previous data indicate that PTP-sigma interacts with TRK receptors, including TRKB. Therefore, we hypothesized that PTP-sigma might restrict TRKB signaling through dephosphorylation. We prepared neuronal cell cultures from mice homozygous and heterozygous for PTP-sigma null allele. We observed that PTP-sigma had a clear gene dosage dependent effect on basal TRKB phosphorylation levels in vitro. We also checked the levels of TRKB phosphorylation in samples from the adult visual cortex of mice heterozygous for PTPRS gene. In line with our in vitro data, adult mice heterozygous for PTP-sigma exhibit increased basal TRKB autophosphorylation in the visual cortex, despite having slightly reduced levels of total TRKB, which may represent a compensatory downregulation due to a long-term increase in TRKB phosphorylation. Finally, we investigated if increased TRKB signaling produced by genetic deficiency of PTP-sigma might render the cortical structures of PTP-sigma heterozygous mice susceptible to plastic changes. We tested adult PTP-sigma heterozygous mice and their wild-type litter mates in an oculodominance plasticity paradigm using optical imaging. As expected, monocular deprivation did not induce an oculodominance shift in adult wild-type mice, but it did induce a shift in oculodominance in PTP-sigma heterozygous mice, indicating that closure of the critical period was delayed or prevented in these mice. Antidepressants have been demonstrated to induce adult brain plasticity through activation of TRKB signaling. They also have been shown to reduce percentage of perineuronal nets in wrapping parvalbumin into neurons, shifting parvalbumin into neurons towards a more immature state and reopening critical period-like plasticity. That's why we asked whether fluoxetine treatment might have an effect on TRKB-PTP-sigma interaction, which could potentially establish a link between antidepressant-induced TRKB phosphorylation and perineuronal nets. We cultured red cortical neurons in vitro and challenged them with 2 different doses of fluoxetine. Fluoxetine dose-dependently reduced the interaction between TRKB and PTP-sigma. We also checked if fluoxetine would be able to disrupt TRKB-PTP-sigma interaction in vivo and observed that acute fluoxetine treatment delivered through an intraperitoneal injection significantly reduced TRKB-PTP-sigma interaction in the mouse visual cortex. Our lab has recently demonstrated that antidepressants directly interact with TRKB through the transmembrane region. Moreover, it has been previously suggested that PTP-sigma interacts with TRK receptors through its transmembrane domain. Therefore, we asked whether disruption in TRKB-PTP-sigma interaction after fluoxetine treatment is potentially mediated by the antidepressant's binding to the same region of TRKB where interaction with PTP-sigma takes place. We transfected hex cells with either a wild-type TRKB plasmid or a TRKB plasmid carrying 2-point mutations in the transmembrane region of TRKB. These point mutations disrupt a cholesterol interaction site in the TRKB transmembrane region, critical for antidepressant interaction. This mutation caused a dramatic decrease in TRKB PTP-sigma interaction levels. However, the interaction was not completely lost, suggesting that multiple sites of interaction may exist. Nevertheless, fluoxetine failed to influence TRKB PTP-sigma interaction in the cells carrying this mutation in TRKB, providing evidence that the site of interaction of PTP-sigma and fluoxetine with TRKB lies within the transmembrane region of TRKB. Finally, in conclusion, I would like to sum up our major findings. First, we discovered that chondroitinase-induced visual cortical plasticity is dependent on TRKB in parvalbumin into neurons. Second, we found that perineuronal nets and PTP-sigma inhibit TRKB phosphorylation, and chondroitinase treatment or deletion of PTP-sigma increases TRKB phosphorylation. Third, PTP-sigma deletion induces critical period-like plasticity at the network level. Fourth, antidepressant fluoxetine disrupts the interaction between TRKB and PTP-sigma, increasing TRKB phosphorylation. Finally, we can conclude that juvenile-like plasticity induced by both chondroitinase ABC and antidepressant treatment is mediated by TRKB activation in parvalbumin into neurons. At last, I would like to present you a working model explaining how chondroitinase and antidepressant treatments promote plasticity by releasing TRKB from dephosphorylating control of PTP-sigma. In the left panel, you can see that in the presence of perineuronal nets, active PTP-sigma dephosphorylates TRKB and suppresses its signaling. In the picture in the middle, in the absence of perineuronal nets, PTP-sigma is inactive and TRKB phosphorylation is facilitated. Finally, in case of fluoxetine treatment, even if perineuronal nets remain present, fluoxetine disrupts TRKB and PTP-sigma interaction, promoting TRKB signaling. Thank you for your attention.